Okay, welcome back everybody. We are going into assignment number 2 for week number 4, which has to do with DNA mutations. And the assignment I have given you this week is to basically take a piece of a DNA sequence, transcribe it into an RNA sequence, translate that into an amino acid sequence. This should all be relatively familiar to you because we did this in Unit 7, which you've already had a test on. We have notes in your notebook on. And so what I'm going to ask you, though, is how when I change something small about the DNA, the bases, the letters, how does that affect the protein that gets made at the end? So what I mean when I say make a small change to something, that's a DNA mutation. And changing the little pieces of DNA sequence can make big or small or no changes on the protein that gets made, and those can lead to us looking different or not looking different. And so that's what we're going to talk about today. All right, so we're going to do this just like we usually do with notes in class. I switch over the camera here. So we got a piece of paper. Normally we'd have our science notebook, but that's okay. This will work just fine. Lock me in there. Sweet. Okay, so the word you want to get stuck in your brain this week is variation. Variation. And what does variation mean? It means variety or differences between individuals in a population. Any population. Now, we always think about humans. So let's talk about humans being different from other humans. But it doesn't just stay with humans. It could be anything. It could be deer, it could be dogs, it could be cats, it could be whales. And the thing is, a lot of us don't notice those small little differences because we don't really see the little tiny tweaks and differences among a whale. We just see a blue whale and there's a blue whale. Well, humans, we definitely notice it. Different shape of the nose, different shape of the eyebrows, different earlobes, way the hair sits on, height, all that stuff comes into play. And this is why, if you've ever noticed when you look at two people who are brothers or sisters or brother and sister, like you can tell they're brother and sister, but yet they don't think they look anything alike. And they're like, how do you know that we're brother and sister? And you're like, oh, it's pretty obvious. But to them, it's not because they don't notice those differences and they don't notice those similarities. They just think, oh, that's just what it is. Okay, so we're going to be talking about how do we look different? How do all the humans, even though we're all humans, how do we all end up looking different? Okay, so there's basically 4 ways that we all become different. So 4 ways to make differences. Okay, the first one I'm not even doing an assignment on because it's pretty understandable. The first one is through sexual reproduction with random partners. Now, when you hear that, everybody always assumes, oh, so it's the same person going around having sex and babies with a whole bunch of different people? No, that's not what I'm saying. That's not what I'm saying at all. What I'm saying is that by, this brother marrying this woman and having a kid, and then this brother marrying this woman and had a kid, you take the DNA from one family, mix it with the DNA of another family, DNA from that same family mixing with a total third family. And so you're getting a mixture of DNA based on the partners and on that. So that just happens, okay? And this is why if, we used to, I wouldn't say we used to, but generations and generations ago, it was like people from a certain part of the world all looked a certain way. Well, that's starting to disappear because as people travel around the globe, have babies with people from other parts of the world, you're starting to get new combinations of DNA that didn't exist before. And so people don't necessarily look from like certain parts of the world anymore. You look at people and you go, wait, are you from over here or are you from over there? I can't really tell anymore. And it's going to continue to get like that. We're going to finally get to the point where we can't really notice certain areas of the globe anymore, and people won't be able to tell where you're from just by looking at you. All right, so that one I'm not even going to get into. That's the first one. Okay. The second one and the third one go with assignment number 3. So that is crossing over in meiosis. And that is independent assortment. In meiosis. So as you can tell, these are both during meiosis. And so you're like, what the heck is meiosis? Meiosis is the process by where you make the sex cells for reproduction. So if you're a male, this is the process by which your cells make sperm. If you're a female, this is the process by which your cells make eggs. And so the different sperm and the different eggs inside the same exact person are completely different than each other. There's millions and millions and millions of sperm, and they are not the same DNA in each one. And so it depends which sperm gets to which egg that makes the combination of you. And so how do you make all those different sperm and all those different eggs? It happens during meiosis. And there's 2 spots in meiosis, crossing over and independent assortment, where it happens. So that's going to be in assignment 3. So after you're done with this assignment, you'll do assignment 3 where you'll watch some videos. There'll be a video by one of our tutors from UCSD, and he's going to talk specifically about crossing over and independent assortment. So pay attention to those. So I'm not going to cover those here. The last one is the one we're going to talk about right here, which is DNA mutations. Put that in caps. So that's the one we're talking about here. DNA mutations. How does the DNA get changed? Because if you change the DNA, then you change the protein. If you change the protein, you could change what it actually does. For instance, if you change the DNA of the gene that controls eye color, you can actually make a different protein that goes into your eyes, and that different color will create a blue eye color instead of a brown eye color. Did you know that blue eyes are a mutation? Okay. Thousands of years ago, humans did not have blue eyes. This is a mutation that happened, I want to say hundreds, if not thousands of years ago, but that's what we've learned. So blue eyes is a mutation. Why? Because somebody had a DNA mutation that created a different protein in the gene that makes your eyes, and it made a protein that now colors blue instead of colors brown or green. There you go. That's an example of a mutation. And there's so many more. There's the one, and we'll talk about these more when we get into genetics. There's 6-fingered hands. And if you didn't know that, there are humans that have 6-fingered hands, 6 fingers on each hand, and some have 6 fingers on each foot too. Or sorry, 6 toes on each foot. And so that actually is mutation. The DNA has changed to where those people are growing a 6th finger or a 6th toe. Okay. Now, you want to blow this up into super sci-fi, you know, you got like the X-Men, and there's like mutations where, like, you know, all of a sudden, like Wolverine has a mutation in his DNA where it makes bones come out of his hands, which eventually turned into steel. That's a whole nother story. But, you know, that's what they're saying in those comics is, oh, this is a mutation. And so the X-Men are mutants. That's where that comes from. Okay, so we're going to go real basic though. Okay, so we're going to get into DNA mutations. So DNA mutations, let's make that nice and big. Okay, there's basically 3 different types of DNA mutations. Okay, and we're gonna make a chart. Keep these all nice and clean with one another, so it's easy for us to keep them separated. All right, let's do it this way. Okay. It's always good to have straight lines. Should have done that in the beginning, shouldn't I? Haha. All right. Let's go. Boom. I'm probably gonna have to double squeeze these in, but that's okay. All right. So let's go here. Let's go. Well, I'll put those names in later. Okay. So this is the DNA sequence in this column. This is the mRNA sequence. And this is the amino acid sequence, which is what you are going to be doing for your assignment this week. All right, so that's our heading. All right, so I'm going to give you the original gene, the original DNA. Okay, so this is like before any mutations took place. And I'm just going to give you some basic sequence. TAC, TTC, AAA, CCG. Okay, so that's the DNA. How do you make the mRNA? Well, if the DNA is a T, you put an A. If it's an A, you put a U. If it's a C, you put a G. Okay, and then TT goes AA. Instead of C, goes G. What matches up with T or A's would be T's, but there's no T's in the RNA, so it's U, U, U. And then CCG would be G, G, C. Okay, so use DNA to make the RNA. All right, from the RNA, we make the amino acid sequence. So how do you do that? Well, you got to have your handy dandy genetic code. So let me slide that up. AUG, I go right here. AUG, methionine or MET. AAG. AAG is lysine or lyse. I'm going to go down here. UUU is phenylalanine or phe. And GGC is glycine. Okay. Now, this is a very short DNA sequence, super short, but just to give you an idea, these make these, and then these lead to those. That would be our normal DNA or our normal gene. Okay, let's start off with a mutation. Okay, so it starts off the same, TAC, same TTC, TTC, but this time, this first A got switched. Something got changed. Now it's instead of AAA, it's CAA. And then the end of it is still CCG. So what got changed? This guy right there. Used to be an A, now it's a C. Okay, what happens when you change one of the letters, but everything else stays the same? We call that a mutation called a substitution. Okay. Think of it like in a basketball game when you take somebody out and the next person comes in, that's called a sub or a substitution. You're substituting one player for another. All right, and then what's the sequence we get in RNA? So still AUG, AAG. Now instead of UUU, it's GUU. And then CCG would still be GGC. Okay, so how is that going to affect the protein? May do something, it may not. Let's take a look. Well, AUG is still MET, methionine. And AAG, just like it was up here, is lysine. But instead of UUU, it's GUU. So I go now GUU, that makes valine. That's a different amino acid. And the one on the end is still glycine. So what got changed? This amino acid is now different than this amino acid. Why? By changing this one letter, I ended up changing this one letter, which ended up changing the amino acid. Now, you might say, oh, does that make a big difference, changing one amino acid? It could. If it's a very important amino acid, it could change the way the whole protein is folded. Or if it's not an important amino acid, it could really make no difference at all. So how important that change is depends on whether it's a certain amino acid. But it did change it one way or another. It did change it. And so your DNA is always going to make this protein. You're no longer making this because your DNA got changed. A mutation took place. So now you're making this protein. You don't make this one anymore. Okay, that's what a mutation is. So a change in the DNA leads to a change in the RNA, which leads to possibly a change of the amino acid. And that could be important, or that could mean nothing at all. It depends on how the protein folds, which is why for your assignment number 1, you did protein folding. Okay, so that's called a substitution. When you switch one in for another one, substitute. All right, now we're gonna go back to the original. I'm gonna give you a different mutation, okay? This time, it goes TTC. AC, just like it did in the beginning, but I'm going to put a G in here. There's no G in there now, and I'm not taking one out. I'm just going to push a G right in there. So now it pushes everything back to the right. So now it goes GTT, which then pushes this C over here, and then AA, which then pushes that A over here, goes ACC, and then that G now, that was on the end here, is sticking off the back. And so by inserting this guy right here, this came in and kind of pushed everything back. So everything got pushed that way. It's like when somebody butts in line and the whole line has to move back for that one person that butted in line. Same idea. But comes in here, pushes everything back. Okay? So what kind of mutation is that? Well, I inserted a new base that wasn't there before. So this is called an insertion. Insert one new base, pushes everything back. And by pushing everything back, you can see it really changes things a little bit here. Okay, if you compare this one up to the original, this is now different, this is now different, and this is now different. So because it inserted here, it changed this, and it changed this, and probably everything after it too. So we go to the RNA. What's it going to look like now? All right, I got AUG, just like it did, but now it's CAA. CAA turns into GUU. And ACC now becomes UGG. And that G right there is a C. Okay, so you have an extra base now where we didn't have an extra base before. Again, compare this one, not to the second one, but compared to the first one. This is the original, this is the mutation. We inserted one, pushed everything back. All right, what does that do to our amino acids? Well, AUG is still met. No change there. Okay, but this is now CAA. So I go over here, CAA is glutamine. And GUU, we knew that one up there. That's valine. UGG is tryptophan. Okay, so now look at this new amino acid. By adding one new base in here and pushing everything back, the only amino acid same as the original is the first one. The last three amino acids have all changed. All of these have changed. They're not lys, phi, and gly. They are now glyn, val, and trip. And so you go, well, what's the whole, who cares about that? Well, now you've got three different amino acids. So now that protein is going to be completely different than the one that was made up here. So when you get something like this happening, maybe this protein doesn't work at all. Or maybe this protein now creates blue eyes where we didn't have blue eyes before. Maybe this protein makes you have 6 fingers because it's a totally different protein now. Maybe this protein allows you to see better. Maybe it's a good mutation. Maybe this protein keeps your eyes from developing properly. So this is a really bad mutation. You don't know the effect until the protein is made. But something is new and different about it. And that's what a mutation means. There's something new and different about the protein. Okay? So substitution, switch them. Insertion, you add a new one in. So we've switched them. We've added a new one in. What do you think the last one is? If you are guessing take one away, that is correct. Okay. So let's look back at the original. Okay, so we have TAC, just like we did. Okay, but this time I'm going to take this, it usually goes TT. I'm going to take that second T and I'm going to pull it out and get rid of it. So the C slides to the left. So now I got T, C, and then the A comes over. And then now these A's shift, so I get AAC. And then now all I have left right here is CG. And so what happened? The T that was right here, it took off. It got squeezed out. This is like when you're standing in line at Disneyland ride and you gotta go to the bathroom and you leave and then the whole line shifts forward a little bit. So this way the whole line shifted forward, slid that way, and because that one left. So how does that affect our mRNA sequence? Well, it's kind of the same deal that we had before, okay? TAC, still AUG, but now TCA is AGU, AAC combines with UUG, And then we have GC. So what does that do for our amino acids? Well, I get out my genetic code. AUG is still met. AGU is serine. And UUG is leucine. And that's it. So this is different and this is different. Now, people might be asking, well, what happens with this guy? And what happens with this guy? Well, they're not three. They're not three bases. So they don't get coded at all. They don't even make anything. So up here, you still only had 4 and an extra, but that extra doesn't do anything. Over here, you have 3 and 2 extra. Those 2 extras won't do anything. So now you only have 3 amino acids. So again, this now looks completely different than that. So this mutation and this mutation really screw things up, whereas a substitution mutation only kind of messed up one thing, okay? And we didn't name it. So I took one out. What happened? What do you say when you're typing something and you get rid of it? A deletion. You deleted it. Deletion. Okay? And the whole thing slides forward. So if you were to look at this, which type of mutation really causes the most damage? It's deletion and insertion. These two really screw things up. Look, that protein at the end is a totally different sequence than it was originally. Whereas the substitution cannot really mess it up too much. It's still basically the same. Yeah, we changed one amino acid, but it's not the same as changing everything like we did down here. Okay? So that is basically your 3 types of mutation. So let's do a quick example. See if you can spot these things before me telling you. So I'm going to give you a DNA sequence. So I give you TAC, AGA, GGA, CCT, AAA. So this is the original. I'll even put that. This is the normal unmutated sequence. All right. So now I say there's a mutation that took place. The mutated sequence is TAC, AGA, GAC, CTA, AAG. So then I ask you, what type of mutation happened here? Okay, so let me see where the difference is. TAC, TAC, that's the same. AGA, AGA, okay, that's the same. So nothing happened there. Okay, this is where it gets funky. Here I have GGA, here I have GA. And then I got CCT, and wait, look, CCT, A, and then now all of a sudden there's a new one on the end. Where did that new one come from? Probably because when it shifted to the left, a new one came in. So if it shifted to the left, that means I must have lost something. Ah, yes, here I have 2 G's, here I only have 1. So this G took off. So when something takes off and the whole thing slides that way, what is that? That's a deletion. Okay? That's what we're looking for. That is being able to recognize the mutation that took place. So it takes a little trickery to be able to look at that and go, okay, wait, there were 2 G's, now there's just 1 G. What happened? Oh, I must have lost a G. So he took off and the whole thing, this whole thing slid to the left, like people moving forward in line. And that's what gets me this right here. So that's a deletion mutation. Okay, so just to recap it, 3 types of mutations: substitution, insertion, deletion. Make sure you feel comfortable with those 3. And then being able to recognize what had happened when I give you this type of situation. So if we were to go over to the online notebook, if you give me 1 second here to pull it, refresh it. Let's go screen share. It's loading it up right now. So this is your online notebook for week 4. Again, just a reminder, if you're struggling with this, office hours are from 2 to 2:30 on Wednesday and Thursday. Obviously, you can reach out to me other times through text messages and Jupyter message. All right, so where is this assignment? So scroll. Assignment number 1 is folding. That's not this one. Although if you look at it, it's very similar, right? There's your sequence. You got to transcribe it. There's genetic code. Similar stuff. Okay, here we are. DNA mutation. So this video is what you're watching right here. There's a simulator. If you want to play around with this a little bit, click on this simulator, and it's just something for you to go to and start making your own mutations, and you get to see how the DNA changes and how the mRNA changes and how the protein folds differently. And it's, you know, it's a thing to just kind of play around with and have fun. You don't have an assignment off of the simulator. It's just for you to mess around with to see how this all works. What do you need to do for this assignment? Okay, so I give you original DNA sequence here, like so. You're going to do the mRNA and the amino acid, just like I showed you on these notes. I give you then a mutated one, mRNA and amino acid, and then I give you another mutated one, mRNA and amino acid, and then 4 questions. How does this first sequence compare to the original? What type of mutation do you think took place? So if you compare this one to the original in this one, is it an insertion, a substitution, or a deletion? All right, question 2. How are the amino acids different? So when you got the amino acid sequence here, how are these amino acids different than these amino acids? Okay, just tell me the order is different, or just one of them changed, or none of them changed even. All right, question 3, compare sequence 2 to sequence 1. Okay, this sequence compared to the original, this sequence, what type of mutation took place? Is it an insertion, a deletion, or a substitution? And then the last question, compare the amino acids. So the amino acid sequence you get for number 2 right here. How do these amino acids different than these amino acids? Okay. Do that all on a separate sheet of paper. Take a picture of it. And that is what you will be turning into Jupyter grades for this assignment. Probably looking at somewhere around 40 points, because this does take a little bit of work. You got to watch the video, understand it, and then do it all on the paper. Again, if you get stuck, come to office hours on Wednesday or Thursday, or shoot me a text message or a Jupyter message, and I will do the best I can to help you out. All right, thank you, everybody. Good luck with your DNA mutations and transcribing.